Explanation first.
Okay, guys, I can skip. So, how are you? Good? Good. So, have you reduced the use of mobile phone? No? Who say yes? Melanie, what about you?
I swear it decreases.
And you? How?
Like travelers. I was like 10 and now I'm like,
And now I'm?
around 6.5, so
6. Okay, so what is the goal?
0.
No, 0 no, amigos.
2, 3.
2, 3 will be okay. Podcast included. And you, Melanie?
5 hours.
Really? From
Yes,
10? Ah,
from
good.
10.
Okay, sometimes the Apple connect the iPad with the telephone. Okay, but it's easy. We just remove the hour that we spend on Canva. But 10 hours, I mean, how long do you stay in Canva? But then I don't understand why you stay in Canva. Because you have to download the material. You know this, next year, all this material disappear. So considering that you are paying a lot, okay, download all this material. Okay, so after this, okay, do you want that I make a review or refresh?
Yes.
Ok, vamos a hacer en español se dice un refrito. Freír is frying, refreír is refrying, un refrito. Okay? Good. You will learn some Spanish words also. Yes? Okay. O un repaso. Okay? Se dice también repasar. Okay, so what we start to talk about on Friday, no? We start to talk about structure of the protein, no? Okay, first question. How many amino acids do we have? 20. Which are the type of lateral chain that we can find in amino acids? The lateral chain can be aromatic, polar, non-polar, charged, and okay, which are the polar ones? The polar, then which are? Okay, no, I don't say this. In this category we have, we say positively charged, aromatic, okay. We have positively charged. In this positively charged we include the positively charged plus, sorry, in the polar we include the polar and positively charged and there is another one. Which amino acid is polar? Which one? Tyrosine. Okay? We have polar. Okay, within the polar, which are the functional group that we find? Hydroxylic group, carboxylic, sulfhydrylic group, and amino group. Muy bien. In the non-polar, which functional group we find? Methyl group or a combination of methyl group. But if in a chain I have CH3, CH2, CH2, SH, how will be this amino acid? Will be polar. If I have CH3, CH2, CH2, NH3, how is polar? Because we said that when we have a carboxylic or amino also in a context in which we have also methyl group, this amino acid will be Polar. Okay, what is the goal? Just to prepare ourselves for the exam. Imagine that we have a polypeptide. We need to say how many amino acids are, how many peptide bonds in general, how many peptide bonds we have, one less than the sum of the amino acid. Muy bien. And then for each of the amino acid, we need to study the lateral chain, and we need to say if this is positive, polar, non-polar, aromatic, positively charged, negatively charged. Okay? So then, okay, when we say the sweeter ionic form, are we able to write this sweeter ionic form? How is it? So, how is it? Carbon alpha? Then, how we write the amino group? NH. Then, and then, muy bien. We call this sweeter ionic form. We have the non-ionic form. How we write the non-ionic form? NH2, carbon alpha, H, then CO, and then the R group. Estamos in how we can consider amino acid. Give me an adjective for amino acids. Or two. They are, all of them are, so, all of them are amphoteric compound, and all of them are how? We say, when we have these two functional groups, they are so in muy bien. Then the solubility will change on the base of the lateral chain. Okay, but remember, all of them are soluble. They are amphoteric compounds. So an amino acid in acidic environment will behave like a base. And this means what? It behaves like a base. They will accept a in a basic condition. It will behave like a means what? That will muy bien. Okay. Do you remember the titration curve? Yes, no? Okay, okay, how we will proceed when we have a titration curve of one amino acid? Okay, first of all, why we do the titration curve? What is the final aim of making the titration curve? To find out which values. The pK. How many pKs we have in the amino acid?
2 or 3.
At least 2, and in some of them?
3.
Okay, why? Which is the condition by which an amino acid has a pKr? Because in the lateral chain we have a group that can accept or give up a proton. Example of this functional group. That can the hydroxylic, carboxylic, sulfhydrylic group, amino group. If in this lateral chain we have one of these functional group, he's smiling all time. Yes, you are. Okay, but it's fine because this means that you are happy. So we need happiness. Yes, muy bien. So, if we have in this lateral chain one of this group, we will have the pKr. Muy bien. Okay, in the case of histidine, why is important histidine? Because he is a buffer system where in physio solution. Okay, so in this case, in order to say which is the net charge of this amino acid through the pH scale, what we have to do? We have to divide. Okay, this will be the first, second, and number 3. Okay, so from this to this, from the 0 to the pK1, how will be the net charge? Plus 2. Lo vamos a poner aquí. In this other range, we are plus 1. 0 and -1. I say pH 12, there will be the net charge -1. pH 7, that is the one that we need, will be 0. Okay, now we compare this pH with the glutamate. Glutamate, which kind of amino acid is? Nega is glutamic acid. So is acid nega polar or non-polar? Muy bien. Now, in this case, we have, again, we divide. In this case, what is the pH of the glutamate at pH 7? 7 will be like this, will be so -1. So the -1 will be at pH 7, glutamate will have -1, while histidine will have a net charge equal to 0. Okay, imagine that in the exam you have this kind of question, you proceed as we do in class, and the answer will be easy, okay? So which organ we need to use during the exam? The brain, yes, and we need to be able to cope with stress, okay? Because when stressed, but I want to say something. Everyone had made an exam, me a lot. Of course. Do you know, I will tell you my experience. In this second degree, I was so stressed with just a teacher. This teacher was, for me, a bad teacher because he asked question, just there are, this letter of the graph, of this value, of this table, all the exam like this. So he forced students to memorize information, okay? This is how the exams were done in the past, especially in the Faculty of Medicine. But there is a total opposite way in which I used to study. I studied to learn, to think, to solve a problem, not to memorize information. This is the only moment in my life in which I was so stressful, and then also if he repeats the question, I was unable to think because I was so stressful, okay? Because I was not confident with this kind of exam, okay? But in our case, if we train every class, okay, and then you study at home, you make this information like yours, when you arrive to the exam, you will be prepared because we are training every day, okay, every class. So don't panic. If you are here present with your body, but especially with your mind, and you will learn the methods, because at the end we learn a method. When you arrive to the end, okay, of course there will be questions that you don't know, but if you're able to think, okay, and you have learned the method, you will pass the exam. I'm sure about this, and you will say, for me the exam was easy. When people start to say, was so difficult, so is because they were not self-confident, so they have no idea how to solve a problem. You have the online test, make them to exercise yourself, find out other quiz in other universities, and make all these quiz in all the languages that you know, in English, French, Italian, Spanish, okay? Also when I was a student at this university, I made tests in English, Spanish, Italian, also French sometimes, you know, because if you train a lot, then the question, guys, will be always the same. The chapter are this one, the topic are this one, so try, they train a lot. Okay, so different type of chemical bond that the lateral chain can form. Non-polar, with non-polar will form which kind of chemical bond? How we call? Hydrophobic interaction. Then, when we have charged, we will have more electrostatic, then we can have a van der Waals interaction, and then we have another type of covalent bond, that how we call disulfide between residues of Cysteine. Cysteine, if you remember, was another amino acid. There is a buffer system in physiological solution. There are just two. Cysteine, different type of, I say that this slide is extremely important. Glycosylation. If I ask you what is the glycosylation, what you will reply? It is a chemical bond between a protein and a sugar. We have two types, the N and the O glycosylation. Why we call N and O? Because N is is the residues of asp, che significa? Aspartic acid has the NH group that will form this covalent bond with a sugar. This is a sugar. And the O glycosidic bond is because the oxygen of the che significa? Serine, okay, is the OH. In fact, if you go back to the slide and go OH, which kind of amino acid is? What do you expect? OH, functional group, will be? Sì, pero ¿cómo es el amino acid? Is polar, non-polar? Okay, go back to the slide, look at the serine and tell me what is the lateral chain of the serine is the CH2OH. Yes, good. I said also in which of the organelles are made this chemical bond. O glycosylation are made where? Just in the plus, yes, so, and N-glycosidic bond only in the RER, che significa? Rough endoplasmatic reticulum. N-glycosylation exclusively in the rough endoplasmatic reticulum, O glycosylation half in the rare, half in the Golgi apparatus. But we will study this better in biology. Which kind of EGG? What is an EGG? Is an antibody. Okay, here we have the formation of disulfide bond and the phosphorylation. Who makes the phosphorylation? We say who makes the phosphorylation? An enzyme called kinase. And the one that remove is called? Muy bien. Peptide bond. Which kind of chemical bond is? Is a covalent polar. Is a covalent bond. How is established? Because we have always an order. The first carboxylic group of the first amino acid plus the amino group of the, how we call this reaction? Condensation, because we lose. Muy bien. The peptide bond is able to rotate? No, is fixed. Who is the molecule, the element that can rotate? Is only the carbon alpha. Cis and trans, we understand when the two amino acids are with the R group on the same plane, we have cis. On the opposite, we have the trans. The dogma of molecular biology, just to go fast, is what say the dogma of molecular biology? That the DNA is translated, transcribed into, and the mRNA is. So if I ask you where is contained the information for the synthesis of a protein? In the DNA. Okay, imagine. I have a doubt if you are sickle cell anemia. Where I will find the answer if you have sickle cell anemia? Imagine that you are a medical doctor. How you will proceed in order to say this patient has or do not have does not have a sickle cell anemia? How do you find? Okay, imagine in the
basis of DNA.
Okay, but imagine that you have no money to see this. First of all, what is how we can see that a person has a sickle cell anemia? In the blood. In the microscope. What we are going to see under the microscope? The blood. Okay, so the cheap way is the microscope. But I will say to you a tip. if you are carrier, do you remember when I explained this carrier? Okay, if you are carrier means what? That you have one copy, there is the correct one, and one copy there is mutated. How will be the genotype of a carrier? How is called the wild type hemoglobin? H? Say HbA. And the wild type, do you remember what is the meaning of wild type? The original correct one, wild type. And the mutated is called muy bien. HbS. How many scenarios we can have in nature? 3. The first one is HbA. This is the second. The third scenario is HbS. Okay, we said, we come back. I say the cheap way to see if we have sickle cell anemia is by analyzing under the microscope the histology. I mean the shape of the red blood cells. How are red blood cells? Rounded. And in the case of HbS, they have this circle shape, okay? But we see a lot of these only when we have both mutated. In the case that we will see in the third lab, we have a carrier, we have the majority will be the normal one, and just a few red blood cells have the circle shape. Why? Because in biology, when we are carrier, we carry the mutation, but the good copy, always tries to compensate. Okay, so the good copy will generate the red blood cells, the normal one. Okay, so imagine that under the microscope, I see just a few sickle cells. This is an indicator that tells us maybe this patient has sickle cell anemia. So what is the next step? Now we will do a genetic, how we say, genetic analysis, muy bien. Genetic analysis, and what we are going to see in this genetic analysis, the gene of the hemoglobin. Do you know how we do this? Considering, with the gene project, the genome project, human genome project, we know exactly the sequence of our DNA. So this means that we know the sequence of the HbA. So with a PCR, we amplify this sequence. Do you understand the meaning of PCR? Polymerase chain reaction. What we do with the polymerase? We amplify one gene. Instead of having one copy, we have 10,000 copies. And then what we do with this amplified region? We make a sequencing. Sequencing means that we are going to see exactly each nucleotide. Then this sequence, we put this sequence in an online page that is called BLAST, that you can see on NCBI. And you will compare your sequence with a wild type sequence. And the BLAST will say there is a 99% of identity except for one amino acid, in the case of sickle cell anemia. In the case that you are okay, how will be the coincidence? Will be 100%. We understand this. I am explaining which are the different steps to conclude that someone has a sickle cell anemia. We start with the histology and then we do the genetic test. Okay, good. What happened in sickle cell anemia? I explained this, no? Which amino acid is replaced with which one? Glutamic acid is replaced by a, okay, glutamic acid, we say there is negatively charged, so it is acidic and polar, and valine is non-polar. So we change one polar with one non-polar. What happens when non-polar amino acids are close together? They form Okay, this hydrophobic interaction, okay, produce agglomeration, formation of aggregates. Understand aggregates? Polypeptide chain that join together with hydrophobic interaction that tend to precipitate. Okay? So this means what? That the people that has the sickle cell anemia, they have a good health in general? No. Of course, the different symptoms will depend on the severity of the disease, but of course, one of the most important symptoms is they feel very tired because of the inability of this hemoglobin to carry properly the oxygen through the organ and tissue of the human body. Okay? Yeah. If you understand this, we'll be fine. Different type of chemicals, structure of the protein. How many we have in total? 4. All the protein have 4?
No.
And until the 3, all of them. And the 4 only which protein? In general, the I capelli? Okay, we are waiting until December to cut our beautiful hair. Yes? I think there is no one. Esto se lo pone detrás. Okay, I don't like any one of your haircut. So I'm talking about the guys.
No. Cheers. Thanks.
You're welcome. Maybe we can save you, but also you need una spuntatina. Ernest, what about you? Your next appointment will be in December as well?
Maybe.
In December, we wait until December, but you can cut by your own, no? Like,
I'll do this. I'll show you tomorrow.
yes,
Please.
tomorrow, yes. Yes, if you do like What about you?
No, I'm a bit scared to get my hair cut here.
Ah, see?
I
Why?
don't want them to mop my hair up.
And you are to wait until
I
December?
don't know, I haven't decided yet. We'll see.
You are going to home after the exams?
Of course.
Who say yes? And how long are you gonna stay?
2 weeks.
2 weeks?
Yeah.
After the exam?
Yeah.
But the exam means which exam? Of November?
November. So that's
Oh no, I'm not.
Oh no,
I'm
I'm
going.
leaving at the
no.
end.
I'm going.
No, I'm leaving in
December.
In
straight
November, who
away.
is leaving in November?
I'm going to pass that.
But just for a weekend? 2 weeks?
Yeah.
And you will miss 2 weeks of class? Because you miss your mom.
Yeah, I'm going to my best friend in France, and I'm going to went to Morocco for my parents. 1 week on.
Con calma. So, 2 semanas in November. And I also want these 2 weeks in November.
How long is this?
2 days because we can.
And with the class
There's a long weekend.
online?
Yeah.
Don't lie to yourself. You won't go
Like,
online.
But sometimes it doesn't work, like today, you know?
After school?
No, but I'm not connecting because I'm not able to remove the session of this girl. See? This happened like today, I put cerrar session, but it doesn't work. Okay, how many levels we have? Primary 1 means what? Sequence of
Amino acids.
number 2, local folding. How many local folding do we have, Ernest?
3, 4.
3 local folding. Ernest is totally wrong because we have the name of these two, alpha, and the tertiary structure means the three-dimensional structure of the protein. Okay, let's say something about the beta sheet. How many amino acids we have for each turn of the helix? 3 points. Okay, which are the chemical bonds that maintain stable this secondary structure? The hydrogen bond between amino and carboxylic group of the first and the fourth, the fourth and the eighth, and so on. Beta sheet, how many types do we have? 2. Parallel. What is the meaning of parallel, anti-parallel? In the parallel, the chain are always in the same direction, mean NH-COH, NH-COH. In the anti-parallel, we have NH. Muy bien, está muy bien. Guys, I'm really happy today with your preparation. Okay, we have to continue like this to be the best group of dentistry, okay? Yes.
So we can take 2 weeks.
We take 2 weeks, of course. We need to be the best ones, okay? Guys, I'm really competitive, you know? So I want to have my students are the best one, okay? So, but in order to be the best one, we have to say, I cut over our hair. Of course, these are the beautiful students. Okay, to be good one, we have to study hard and to remove on our telephone
5 hours.
TikTok and Instagram just 2 weeks before the exam, and you will pass everything. You know this because TikTok is killing answers. But if you repeat this as mantra, you know what is a mantra? You know what is a mantra? No? Nobody knows what is it?
Like a charm.
Something personal, like ritual innit?
Something that where we use the mantra. For example, when we do meditation, yoga. Okay, I have a song if you want. That I put when I cannot sleep, and this helped me to relax. Mantra is a sentence that is repeated all the time, okay? So, and you have to repeat in your mind this mantra. TikTok is killing my brain because it's killing and my neurons cannot form, and this means Alzheimer's disease in 20 years. Yes? Chicos, it's science. I'm not inventing anything new. Okay, alpha helix can be, how we say, left-handed and right-handed. Okay, we stop where? ¿Cómo se llamaba esto? The motive. Which motive we mentioned last time? Zinc finger and
Helix, turn helix.
muy bien. Where we find the helix, turn helix and zinc finger in proteins that vamos a apuntarlo bien. In protein that binds the DNA. They are transcription. What is a transcription factor? A protein that participates in the transcription process. Muy bien. We stop here, isn't it?
Yes.
Okay. When we say protein, how many types of protein we have? Categories of proteins. We say fibrous protein, then read. What is the meaning of transmembrane protein? A protein that is in the membrane. We say embedded in the plasma membrane. Okay, my friends, the amino acid that are in contact with the phospholipid, how they will be? With the details. How will be this amino acid? Muy bien, non-polar. And then inside, depending of the molecule that pass through, we can have polar or non-polar amino acids. Muy bien. Protein structure. Okay, quaternary structure. We have the quaternary structure only in some proteins that they are formed by different peptide chain. This polypeptide chain join together to form, in the case of hemoglobin, a globular protein. In the case of hemoglobin, what is important in the structure of hemoglobin? We have 4 chain, 2 alpha and 2 beta chain. We have the heme group in which we have a how we call this? Iron. Why is important iron? Because iron will bind the oxygen, okay? In the heme group, then now we will study more in detail, we have the iron. I explained the concept of misfolding, folding related to disease.
No.
No? We are right here?
No.
Melanie?
No.
No? Muy bien. Neurodegenerative disease. This is what can happen to us if we continue to stay 10 hours on the mobile phone. Neurodegenerative disease. Okay, I don't know when it was, maybe yesterday in the news, there was a girl, a woman, okay, that has Alzheimer's disease since 20 years. She's like 60 and 60 or 65, and she got Alzheimer's 20 years before. So we are talking about a girl with 45 years old. And one of the causes that can also, increase the onset of the Alzheimer's disease is because she was lonely. Okay, she has actually 2 daughters, but maybe they do not have any relationship. So this girl spends the majority of her time alone. And when we are not relationship with other people, something that is happening because we stay home with the mobile phone, we think that we have relationship with people, but actually no. This means what? Imagine, you live with these 2 guys. So every day you have to discuss, to argue, to say, okay, you have to do this. So when we stay with other people, we need to solve everyday problems. Okay? And this also helps us to think, to create this neuronal connection. Okay? So this is one of the factors. So that's why I say when we are healthy, when we are actually happy, yes, because do you know what is the definition of health from the ¿cómo se llama? La Organización Mundial de la Salud? What is the definition of health from the ¿cómo se dice? World, la Organización Mundial de la Salud. ¿Cómo se dice en inglés? World Health Organization, eso es. WHO. Okay, let's see what is the definition of health, because this definition changed all along years. Do you find it? Read them. Your name is
Anna.
Anna. Just with one N or with
2 N's.
Anna?
Yeah.
I like Italian. Anna. Okay, and you pronounce? How do you pronounce?
Anna.
But it looks like with the one, no?
No.
Anna.
Anna with 2 N's, yeah.
Vale. Anna. Say. Okay. Read.
Would you read it out loud?
Yes, of course.
Health is a state of complete physical, mental, and social well-being.
¿Ves? Que say mental, physical, and social. Continue.
And not merely the absence of disease or infirmity.
Absence of disease or
Infirmity.
infirmity. Okay, so this means what? That we consider, as a future medical dentist, you need to have clear the definition of health. That in the past, health was the absence of a disease. Okay, this definition changed, especially in the last 20 years. But now we say that a healthy person is the one that has mental, physical, and social. Okay? So let's think about your life and say, okay, I'm okay, or maybe I have to work on this. If we have mental problem, what we have to do? We go to the psychologist. We have money to do this. Yes? I mean, in this class, I can say this. If you feel stressed with anxiety, that you cannot cope with all this stress, don't take any pills. Go to the psychologist or just have a walk. 10,000 foot every day and you feel very bad, much better. Yes, okay. Now, neurodegenerative disease. The ones that you know are the Alzheimer's disease and then the Parkinson disease. We have MEDCO, or this one, there is a Creutzfeldt-Jakob disease. Okay, so what is the meaning of folding and the misfolding? And what these have to do with this neurodegenerative disease? A folding of the protein is the 3D dimension. And how is this three-dimensional structure? Is the correct one. And to this correct three-dimensional structure correspond a protein that is functional. Means structure is related with the function. Correct folding, correct function. Misfolding, we understand misfolding is when a protein does not assume the correct three-dimensional structure, and this produce disease. Example of this neurodegenerative disease, Alzheimer's disease. What we have in Alzheimer's disease, guys, we have in this slide, we have an accumulation of amyloid beta or beta amyloid is a protein. Okay, is a misfolded protein, and we have an accumulation of tau protein. In the case of tau protein, we have a non-correct phosphorylation. Okay, tau protein. In the case of Parkinson disease, we have also an accumulation of amyloid, tau protein, and alpha-synuclein. Alpha-synuclein is a protein, if you can annotate, that join the elements of the cytoskeleton. If I remember well, will connect intermediate filament with microtubules. I think so, okay? Just in case, double check. And then in the MEDCO disease, we have the prions. Have you heard about MEDCO disease? You are from England? This happened, I don't know, maybe 20 years ago. I don't remember. Okay, look, find information about MEDCO disease because it was was a problem that happens, I think that, I don't remember, in the 19 or in the 20.
80 to 90.
80 to 90, and it consists of MEDCO disease. En español o en italiano es la vaca loca. La vaca loca, ¿qué significa? What happens? Read the story.
Wait, hold on.
Okay, so we made some story of the disease.
Fatal brain disease.
Something that is reduced. So
It's a fatal brain disease in cows.
So is a neurodegenerative disease in cow. And this happened because of what? Because imagine one cow has the disease. And you know that in industry that produce the feeding for animals, they use this cow to generate feeding for the other one, and this is how it spreads. Okay? So the mad cow disease affects exclusively animals, and the ones that can affect human are these Creutzfeldt-Jakob disease. Now let's understand the alternative three-dimensional structure. Okay, we can have an alteration in the three-dimensional structure when we have a mutation at the level of the DNA, like in the case of Silker cell. Inappropriate covalent modification after its synthesis. Remember, the synthesis of the protein takes place in the cytosol, but then the modification of the protein means phosphorylation, glycosylation, addition of methylation and so on, continues in the rare and in the Golgi apparatus. In one of these steps, we can have a problem. And if we have a problem, this means what? That this protein cannot assume the correct folding. And if the protein does not assume the correct folding, the functionality will be compromised. Then we have unidentified reason. The consequence, loss of normal function, or marks its proteolytic degradation. Okay, annotate here is via lysosome or via proteasome. What is the meaning of protease? Lysosomes are organelles involved in the elimination of macromolecules, different types of macromolecules, lysosomes or proteasome. The proteasome we will study in biology. You have to think the proteasome, do you know what is a grinder?
Yes.
Okay. Is the grinder for misfolded protein or for old proteins? Okay? Imagine the proteasome like a grinder because it's a grinder, okay? Now, lo degrada, okay? So the grinder will cut the protein in many amino acids in order to degrade the protein. Of course, if the body recognizes that this protein have a mutation and degrades, this will be good for us. But if the body does not recognize this protein as mutated, this of course will affect the functionality. If the degradation of the protein is not complete, we have the accumulation of this protein in different parts of the body. Of course, we don't have only neurodegenerative disease, but we have other diseases. For example, my father, 5 years ago, he started to felt very bad, he cannot breathe properly, and then they discovered after many exams and so on, they have amyloidosis, but not in the brain, on heart. So imagine heart full of this amyloid protein, misfolded protein that the body cannot eliminate properly, and they accumulate on heart, and this impedes the correct functioning of the heart. In fact, my father has a severe insufficienza cardiaca. How to say in English? It's a heart disease when the contractility of the brain is affected. Okay, imagine. Okay, when you will study the heart, the percentage of functionality of the left ventricles that expulse the blood to the periphery. Okay, this percentage is called ejection volume. Is around in a normal person is around 40-60%. My father has like 16. Yes, guys, 16, and then he had to took, I don't know if maybe 10 pills, because you have to increase the contractility. He also has arrhythmia, so you have to take pills for arrhythmia, pills for avoid the coagulation, also the pills from amyloidosis, pills that avoid accumulation of uric acid. You know this? So our life changed completely. Okay, completely because of a mutation. I mean, it's a protein, misfolded protein that affects, in the case of my father, heart. This amyloidosis can affect also the kidney, the liver, or can affect liver, kidney, heart, and also the brain. Amyloidosis has a good treatment, okay? It's very good. I mean, we are okay, but life, remember, can change from one day to another. So, this means what? If we have accumulation of this misfolded protein, this can produce neurodegenerative disease or other diseases that affect the functionality of of different organs. Native conformation, and highlights this. Native conformation is the correct three-dimensional structure. Okay? So where we find the information for the synthesis and the structure of the protein are always present in the DNA. In the DNA, we have the precise instruction to synthesize a protein. Okay, in the cell there are proteins that help other proteins to assume the correct folding. How we call this protein? Chaperones, okay? Highlight this because this we find also in biology. So if I ask you chaperones are proteins that help the other protein, the nascent protein, to assume the correct folding. Okay, how we say when the protein lose the correct folding? We say that the protein is denaturated. Denaturation means the loss of the correct folding, and it can be generated by different factors like high temperature, change in the pH, or presence of chemical agents. Okay, so the denaturation is a process, can be reversible or irreversible, and this depends on the type of the perturbation and the time of perturbation, the type of perturbation and the timing, okay, of the perturbation. Okay, heat. The heat can break the hydrogen bonds. The pH change and also breaks the hydrogen bond by modification of the ionization state and the chemical change like ions and electricity. Okay. Is reversible or irreversible? We say this. And here, guys, we have like a slide that resumes everything that we say until now. Important. Native conformation, the normal protein, different level of organization. Remember all the chemical bond in primary, secondary, tertiary, and possible in the quaternary structure. Okay? Concept of folding, misfolding, denaturation, who generates the denaturation, what happens with denaturated protein in normal condition are eliminated, in non-normal condition there is an accumulation that can produce neurodegenerative disease. Okay guys, so we stop 5 minutes and then we continue. I need to go to the toilet. 5 minutes. Do you have class after?
Yeah. Sorry.
Yeah.
Yeah.
J'ai perdu 170 € hier au casino en ligne.
Are you prepared?
I'm trying to.
C'est même pas casino en ligne, c'est les jours.
Huh?
No, it has.
But it's like a crossword riddle.
Yeah.
I mean,
I'm tired.
Did you watch the movie?
No. Did you?
It's a nice movie. Well, nice. It's like, it's okay. It's not bad.
But you have to pay, no?
No,
What?
I watched it on YouTube.
No, it's that's a trailer. That's not the movie.
No, you can find it on YouTube, the full movie. So it's poor quality.
Yeah.
You can watch it.
Yeah.
Or if you have Amazon Prime,
Yeah.
it's on there too.
Yeah, we tried, but then I can't pay. Like, he asked me to like enter my password over and over again.
Moi aussi, j'ai oublié R.
Moi j'ai pas pris.
Ils sont carrément obligés de te lever pour le laisser passer. C'est bizarre, tu te lèves pas. Hein? Quoi? Tu te lèves pas hein, pour tout le monde, c'est bizarre.
Mais moi je vais marcher le stage. Ah là là, mais c'est où?
Pour laisser passer l'italien, et ben quand tu fais le c'est
Ça va, il passe. Parce qu'il allait dire ce qu'il allait dire.
On a fait encore. Non, on a fait un peu de tour de Madrid.
T'es dans le parc et tout?
Ouais, je suis allée avec Mignon.
On a en vélo, mais je veux dire, on se connaît les filles, il rigole. Il est en train de
Je sais pas, c'est quoi là?
tu nous fais des...
Bah, si, elle est dans la bourse, elle regarde ce qu'elle a.
J'allais dire, pour nous calculer.
C'est quoi là?
Le gars, c'est pas ça. Imagine, j'ai perdu de l'argent, je fais exprès.
Mais non, c'est la bourse qui a laissé.
Bon,
Ok.
Elle a dit, j'ai trop peur de la main. Imagine qu'elle perd tout le temps, j'étais pas pour moi.
Moi j'ai vraiment peur de la masse parce que j'ai pas révisé. En fait, dans ma tête, il allait avoir que la version qu'ils ont envoyée sur
Mais gros, je te jure, j'ai trop la haine.
Mais meuf, j'ai appris ça se passe.
Ah, moi aussi.
Non, ils sont dans un sujet.
Et toi, par contre, t'as fait tes sons sans moi. On a dit, on allait faire des sons ensemble.
Mais comme ça, je te fais des papouilles.
Moi, je te fais la même couleur que Anna.
Ouais, trop cool.
Oh là là, je l'avais même pas remarqué.
En plus, elle m'a dit, regarde les sons de Anna avant de les faire.
Bah oui, parce que non, mais c'est jamais vu.
Je reste.
pour ta gueule.
T'as copié. Elle me l'a dit.
Je suis archi choquée.
Ton comment ça s'appelle? Non, mais tes parents et tout, c'est ça. C'est marrant aussi.
Ouais, la 4 euros, je l'ai tryhard pendant 6 heures. 6 heures sans m'arrêter à la musique. Je suis la seule.
Ouais.
Non, mais en vrai, ça va, ça veut dire. Mais gros, c'est pas le même. Mais tu sais que moi, je suis dans la même.
C'est Ernest.
Quoi? Quoi encore? J'ai rien fait, je suis en train de discuter, on m'engueule. J'ai discuté de t'aller tranquille, voilà.
Et en fait, on va rentrer du boulot, genre vers minuit 30.
Il a pleuré. Je l'ai spoilé.
J'attendais et tout. Et n'importe quoi, il a vraiment
Ah, ok.
pleuré. Genre, il ouvre la porte et je fais ça, tu vois. J'ai mis coquillage.
La salle.
J'avais vu le câlin. Quoi, tu vois.
Oh oui.
C'est mignon. Super heureux. Avec un concert. Et du coup, voilà. Et le lendemain, on est parti en métro, c'était trop bien. On a discuté avec mon père.
C'était que avec tes parents et tout ça.
Non, avec lui, ma nièce, c'était ce star de avec Star.
Awesome.
Like what?
Like a sort of things.
Yes.
Here?
Yeah.
I don't smell anything.
You don't.
Like, no. I think I'm sick, but like, I just don't smell. What does it smell like?
Like the sort of thing like you light up to like get to repel flies. You know those things, candle-like things.
Yeah. Oh, the citron, like citron things.
Yeah. You don't smell that. You don't.
Don't smell anything.
I'm gonna go for more.
I might be hallucinating.
Like it smells like lemon.
Yeah. Yeah, it does.
Don't smell.
Toi, t'avais pas trop compris, je crois. Bah oui, c'est ce qu'il faut.
Les gars, il faut que je regarde les films d'entrepôt.
Tu sais que moi j'ai regardé ça.
C'est bien, j'ai regardé ça.
C'est qui tu peux savoir? Mais parce que pas faire autre chose.
Moi j'ai regardé
Si son lien il marche pas.
Le moulin il marche pas.
T'as regardé les films?
Oui.
What a romantic.
Yes.
Es romántico, Ernest.
Ça,
Oui, romantique.
Do you have girlfriend?
Non, I don't.
Et Carole, il y a extrêmement.
Girlfriend.
Boyfriend. You can say. I don't have friend.
Les gars, je voulais apprendre.
Oui, bah moi non plus, c'est bon.
¿Te gusta algo la chica?
Sí, me gusta mucho. Siempre.
Mélanie?
Siempre se queda.
C'est moi, lui de la.
Es le que hablo con todas las chicas de la clase.
Sí, está muy bien. Es un chico que tiene más empatía.
Es simpático.
¿Cómo se dice? Tú has empatía.
Empathique.
Non, je crois pas. C'est quoi la partie de vie? Non, mais faut pas que je parle à Aimé.
Ah, ça s'appelle chat.
Which class do you have later?
Anatomy.
With Sarah.
See.
Do you like Sarah?
Yes.
But you prefer Sarah to me?
No.
No?
No.
Do you prefer Sarah?
Not over you.
La reine.
Ayer, the queen.
Yes.
Yes.
La universidad.
I'm stressing out.
Yeah.
Anatomy.
Yeah.
Ah, il y a 4 minutes. Voilà. Mais comme
J'en ai besoin du bonus.
Ouais, moi aussi, mais j'ai eu des connaissances.
Bon, parce que j'ai vu.
Yeah. Think about something else.
Yeah. I mean, I look at the time. I'm like, it's like 15 minutes left, basically.
Certainly, like, we could at least like have our own time tomorrow. Because like
Yeah, true. We have like unit 4.
yeah.
You got 4. What? How long have you been so far?
Are we ready?
Zero sugar.
Now why you are sharing this bottle of water every day?
I refill.
I share that.
I forgot my bottle.
But you are like a family, you live together, no?
At this point,
Yeah,
basically.
it is quite basic. Yeah.
How is going in this family? It's great. I think he has no the same idea.
Me? Nah, it's cool, it's cool.
It's good.
Yeah.
Yeah. I'm not take care of you.
No.
No, this. Amor, la puerta. Okay, guys. Let's go back to our beautiful biochemistry. ¿Qué estás haciendo con el
Mais le... Mais le...
Okay. Remind me now? No. Vamos. Okay, classification of the protein according to the function, shape and solubility, nutritional basis, and in terms of structure. Okay, let's go. I like this classification because some of these examples we have already done. Yes, when we say structural protein, we refer to the collagen keratin. Structural protein that have also this contracting function are actin and myosin in our. Have you training the goodbye muscle? You know which one is?
Tricep.
I might know this.
It's not. I train very hard. You know. Do you see?
Wow.
I have how to say hematoma. Look, because I train with the ring. Yeah? It's very painful. The ring. I make the ring for dance, yoga every week. But I don't know if I'm able to do the ring again this week. Or maybe I have to wait. After trying the ring, I say pole dance is so easy. Because ring is very, very... you have to make up on the ring.
Is it like the two-handle ring?
Yeah, yeah.
Sì, sì.
Olympic one.
Yeah.
El big one. Okay? Very hard. A ver.
Oh, that.
El ring. The ring. Esto.
Ah, no, not this one. So a big one. Only one. Oh, it's not the same.
But then, with this kind of activity, you train the entire body, especially the up. I really hate doing up. But with this one, I have strong up. And then, okay, move on. contractile muscle, transport, we know hemoglobin, lipoprotein, the lipoprotein that we study, that we say the good and bad cholesterol, how we call them. I'm not famous. Then I say when you do blood analysis, we do what? We look at, muy bien, LDL and HDL, storage, casein and ferritin. You know that we can have different type of anemia, no? We can have a low level of hemoglobin, but also we can have a low level of ferritin. Ferritin actually is what? The protein that store the iron on the spleen and liver. For example, in my case, I have normal level of hemoglobin, but I have low level of ferritin. And do you know why? Because I don't eat red meat.
Why not?
Hormone.
Why not?
Because I don't like. I made
You don't like steak?
a vegan ragu with no.
Yeah.
No, guys,
It's like
was
same
good.
taste. No protein.
No,
Yeah,
the
you have
protein of
protein.
soy. You mira. Soybeans.
So it's not good for It's not good for hombres.
Claro que es bueno para hombres. Yo tengo un hombre en casa que pesa 110
Yeah,
kilos
but
y le va bien.
if you
Ya te lo digo.
Non, non, c'est pareil.
No,
C'est de la
because
protéine.
it's
de la protéine
green
végétale.
protein,
Yeah,
guys.
green.
It's soy. Soy.
Mhm.
Soy texturizado.
you have to lower the hormone of the male.
Look, if I say this is meat, you believe.
No.
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 No, you cannot believe that.
Yeah, you're veg, you don't eat meat.
Chico, es igual que la carne. Pero estaba muy bueno.
Yeah, but
If you want,
soy can fertilize men.
es que estaba bueno. Today I'm going home to eat. We are so beautiful.
Ça diminue le taux de sperme. Mais c'est une vraie information.
What?
Soy reduce the level of sperm in men. So it can fertilize.
Yes, my love. But you don't need to eat every day.
Yeah, I know, but if some people eat every day.
Soy also can interfere with the metabolism of thyroid. But of course, if we reduce one day red meat and we substitute one day with soy, soy has a very huge amount of protein. Yes,
But chicken is still better.
amor, but it's not. What is the problem of this world? That we cannot eat meat every day because it's not sustainable.
Oh.
That's why. Okay? So, let's try new things. You will say if they are good. And my soy ragu was and is very good. And today I'm going to eat. Yes? Okay. Protein can work as hormones like insulin, growth factors, other hormones that you know. Hormone, you say? Testosterone, estrogen, progesterone, and so on. Enzyme, we know which they are.
LDL and HDL cycle. What is cycle? LDL and HDL.
For the planet.
What does it stand for? LDL and HDL.
¿Cómo es? High density lipoprotein, low density lipoprotein. Low density, high density. Okay? So we have enzyme, immunoglobulins that work in the immune system, and other protein that work as cell signaling. Do you understand the meaning of they transduce the signal inside the cell? Okay. We will study this in biochemistry, but I will introduce. When the insulin binds its receptor, this binding the insulin receptor activates inside the cell a cascade of events. a cascade of reaction that at the end produce a biological response. This cascade of chemical reaction is called cell signaling. Okay? But you don't need to know right now. Okay, guys, ¿dónde estamos? In terms of structure, we have simple protein, conjugate protein. Example, conjugate, when we have glycoprotein, glyco means with glycoprotein, protein with sugars. Lipoprotein, protein with nucleoprotein, protein with nucleic acid. Metalloprotein means protein that have a metal in the structure. Phosphoprotein means with phosphate. Hemoprotein that have the hemo group. They can be complete or incomplete on a nutritional base. Complete, do you know the meaning of essential amino acid, non-essential amino acid? Essential amino acid are those amino acids that we exclusively ingest with the diet, with the food. Incomplete means no, essential, non-essential. Okay. Incomplete means incomplete protein that are deficient of one or more essential amino acids. Okay? Essential amino acid means amino acid that we ingest exclusively with the because our body is not able to produce them. Non-essential means that our body can produce. We got this? Yeah. Then in three groups, I say this before, fibrous, globular and membrane proteins. Okay? Example of membrane protein are all the receptors. What is a receptor? It's a protein that bind up
Neuron.
a
Neuron.
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 Che disse?
Neuron. N-E-U-R-O-N.
No, amor. El neuron is una struttura cellular. Is a cellular structure. A receptor, mira, how we call in general. A hormone, a receptor bind a something that bind the receptor, how we call in physiology?
Signal.
Hello, good morning. Hey. Ligand. Ligand is a molecule, can be a hormone or another protein that bind a receptor. The binding ligand receptor produce a response that can be physiological, chemical, biochemical, biological. We annotate this. Yes, ligand, como ligando, ligando. En español, ligando. Ligar en español is flirting as well. Yes, cuando dice en la discoteca, nuestro amigo estaba ligando. Yes, lesson 1 of Spanish for dummies. Okay, your name? Zach. Sábado de noche estaba ligando en la discoteca. Nice.
Sweet.
It's okay, good. Let's move to the fibrous protein. Guys, it's a little bit boring, this part. I think that you can read by yourself. Okay. ¿Tú qué dices? Okay, fibrous protein. We have to study just 3 types: collagen, keratin, and fibroin. Collagen, alpha helix, keratin, I think there is alpha helix as well, and silk fibroin is beta sheet. Now let's see. They are structural protein. Okay, if I say collagen, what do you say? It is a fibrous protein, but is also a structural protein. Okay, where we find, for example, collagen? in the tissue, and in the tissue, okay, to form the fiber. Okay, muy bien. Now, what kind of amino acid do you expect, considering there is present, for example, in our skin? How do you expect them? Piénsalo. Okay, we make an experiment. We can use the water, family water. Okay. Now, what?
It's water repellent. It's
Okay.
laminar granule.
Oh, so tranquillo. No, stop. So, how is the collagen? How are the amino acids?
Water.
So, by these small experiments, we can conclude that the structure of the amino acids that compose the collagen are non-polar, because if they were polar, this means what? That our skin will solubilize. So, muy bien. In this way, you will remember for the rest of your life. Okay, so keratin. I don't know why we study with keratin. Doesn't matter. It's strong, inextensible, insoluble, and chemical inert. Inert, ¿qué significa? Que no does not react. Muy bien. We find where we find keratin. Skin, nails, and different parts of animals, okay? It is an elongated alpha helix, and this is right-handed. For each protein, make like a resume with the principal characteristic, okay? In this way, you will have this information. Is also present on hair. Then at home, and you say to me next time, find information regarding the oxidation and reduction of keratin in our hair, because if I don't remember well, if we have this cysteine reduced, then maybe we have curly or the opposite hair. I don't remember, okay? But this is a question of oxidation and reduction of cysteines, okay? Look, keratin, alpha helix, now two chain oriented in parallel. Then first we have one helix, second two chains, then we have a protofilament and protofibrils, okay? For protofibrils form 32 strands of keratin. Keratin, very important highlight, it is one of the proteins that form the intermediate filaments. Intermediate filaments are elements of what? Of the cytoskeleton. What is the meaning of cytoskeleton? The skeleton of the... How many are... So, in the skeleton of the cell, we find what? Intermediate filaments, microtubules, and microfilaments. Okay, but we will see later. Silk fibroin, where we find in tissue that have silk. Yes, easy, no? How is silk? It's very flexible. How is it? It is very strong. Okay, it's flexible and extensible. Yes, but when you try to extend silk, it's not possible. We find in silk cloth, okay? In this case, what is the difference with the keratin? In this case, we have, which is the structure of this protein, has beta sheets, okay? And how are these beta sheets, guys? Anti is rich in ALA. ¿Qué significa ALA and D? Alanine and glycine. How they are these amino acids? Non-polar, and they are stabilized by hydrogen bond and van der Waals, as always. Now, collagen. How many collagen I say last time? I say 10, 12 I say? I don't remember. Maybe we have 12 type of collagen in the human body, but more than 20 genes have been identified. We understand the difference between genes and protein. In theory, we have 20 sequences, but only 12 have been discovered, okay, or identified. In human, at least 19 different collagens, because there is the most abundant protein in the human body. The most abundant is type 1, and in the oral cavity we have type 4. Here you have. Of course, do you need that we remember this? No, but type 1 you have to know because it's the most common. Type 4 is also present in the basal. Do you know what is the basal lamina? Okay, esperemos. And also in the periodontal ligament, we have type 4. Okay, you have to remember this. And now we go to the assembly of the collagen. Okay, pay attention. We have 3 different chains, black, red, and green. 3 different polypeptide chains with a unique amino acid composition. We have a repeated sequence of glycine, amino acid X, amino acid Y, GlyXY. This X corresponds to what? X is a proline, and Y is usually a hydroxyproline. All of them, if you remember, they are non-polar. If you don't remember, you go back to the first slide and you double check. So we have 3 different polypeptide chains. Stai fermo. In this polypeptide chain, we have glycine, proline, hydroxyproline. Okay, always this structure. Glycine is located in the interior, proline helps to produce a helical formation, and then we can have many modified amino acids like 4-hydroxyproline. We understand Can you reduce the noise? Thank you. Jesus. Your mom sent you here and I have to deal with you every day. Yeah, your mom now is very happy. Yes? Yes. Okay, now the teacher with the kids. Okay, so 4-hydroxyproline means what? That 4-hydroxy, che significa? Hydroxyl group is in the position 4, 5-hydroxyproline in position 5. Okay, now let's see the assembly. Okay, collagen has a triple helix. Triple helix because we have 3 polypeptide chain. Okay, each polypeptide chain has a left-handed helix. Imagine, no? We have 3 different polypeptide chain with a left-handed, but when they assemble together, okay, they create a right-handed. Yes? Sorry. Okay, eso es. 3 polypeptide chain assembly themselves as left-handed, but then when they assemble, they create a structure that is right-handed. We are talking about a alpha helix. Okay, which kind of chemical bonds are established? Again, hydrogen bonds that contribute to the strength of the collagen. Okay, now for the structure we look at this. Okay, first of all, where we are in the cell. 3 polypeptides combine together to form the triple helix that we call pro-collagen. You have to know this, pro-collagen. Number 2, what happened to the pro-collagen? We cut the extreme ends to form the tropocollagen. Many tropocollagen fibers join together to form the collagen fiber, and then many collagen fibrils form the collagen fiber that have a size of 10, how we say this, 10 micrometer. Okay, I repeat, 3 different polypeptide chains form the pro-collagen. The pro-collagen, to the pro-collagen we cut the extreme end to form the tropocollagen. Tropocollagen join together to form the fibrils, and then the fibrils join together to form the fiber. Okay, you have to know by memory this. Okay, I'm sorry, but okay, now we have collagen structure illustrate the relationship between amino acid and two-dimensional structure. Okay, have you heard about osteogenesis imperfecta and Ehlers-Danlos syndromes? Maybe some of you can suffer from this disease. Many years ago, I don't know if 5 years ago, one student discovered they have this Ehlers-Danlos syndrome. That is characterized by weakness of the connective tissue. If you do something like this, let's do it. If this is, let's do it. Guys, why you are so this? Doesn't matter. It's not a disease that you will die tomorrow. Do like this? No. Maybe you. Guys, I mean, if this, no, here we will do. If this is more weak, this is depend, let's do it.
C'est élastique.
El tuyo es como más...
Oh, that's not
Ah, mira, ¿ves? Eso. ¿Ves?
Do I have it?
Non, non, non.
No, look at it.
Non, mais fais de l'autre côté, l'autre bras, l'autre main.
C'est fait.
Oh my.
This is...
Well, I missed it.
Okay, but it's fine, don't worry. Your life is safe. Nothing, guys. It is a mutation in the sequence of the collagen. You can also see in the other parts of the body. And these are the connective tissue is more weak, and it is related to fragility and skin hyperextensibility. Okay. Then osteogenesis imperfecta means what? That the synthesis of the dental tissue is also compromised. There is a mutation in type 1. This, of course, osteogenesis affects the bones. Dentinogenesis affects the teeth. Okay? You can see some picture like this when you have this gray that situation that you will see in your beautiful career as dentist. And then we have no genetic disorder. Yes, now when you will study this part, you will say, okay, we can have genetic disorder because we have mutation in the sequence of DNA, but we have also non-genetic. For example, the scurvy. This is a deficiency of vitamin C. What is a vitamin C? Un momentito. When we talk about enzyme, we say what? That enzyme needs coenzyme or cofactor where inorganic elements, coenzyme where organic like vitamins. Do you remember this? Now we put this info together. why we need vitamin C and what have to do with this scurvy. Okay, please listen. The scurvy is a non-genetic disease. Why is important the vitamin C? Because the vitamin C is a coenzyme of the enzyme that synthesizes the hydroxyproline. And where we find the hydroxyproline? In the sequence of collagen, glycine, proline, hydroxyproline. So what happens? Imagine your oral cavity. You don't eat anything with vitamin C, so the synthesis of this collagen is affected. And what happens with the scurvy? The scurvy, how we start to see when our gingivas are bleeding. Okay, so with this 5 is clear? I repeat. What do you repeat you? What you have understood? Guys, this chair is not proper for a queen like me. Actually, it's broken. No, dilo en alto. Bueno, yo te corrijo.
It's like vitamin C, it's cofactor with hydroxyproline.
No, it's a cofactor of the enzyme that synthesizes the hydroxyproline. Hydroxyproline is an element of the collagen because the chain of collagen is made by glycine, proline, hydroxyproline. So if we miss one component, what happens? This collagen, how will be? Is impaired, okay? And this is how we can see this problem in our oral cavity because our gingivas, how do we say it? Gum, sorry. Our gums start to bleed. Okay, now we make a small research regarding the scurvy and put on ChatGPT story of the scurvy because it's very nice, but I'm sure that in anthropology you are not going to study. But for me, it's really interesting the story of the scurvy. Scurvy, aquí lo ponemos. Guys, I have to put the sound in my email because what say the ¿Qué dice la historia del scurvy? Anna.
I don't have a story, it just says that it was on the plane.
Guess what we are doing? What we are doing? Here we don't do other subjects. If you have to do other subject, you go out. ¿Estamos de acuerdo? Okay. Say.
It just said scurvy was the scourge of sailors for thousands of years. An estimated 2 million.
Espera, repítelo. Es que ella habla muy bien inglés. So, eso es el problema. I will read with an Italian accent. Okay. You understand better my accent? Es que dice, was, okay, the scourge of sailors. Sailors que son los marineros. For thousands of years. An estimated 2 million sailors died for disease. Find the cure for scurvy article in the Naval Structure website. So, this, okay, when starts navigation? When people want, okay, have you heard about the route of the silk? Have you heard about this? Okay, I will tell this story because it's so interesting. Okay, so imagine that in the Middle Age, the fridge, as we know, like today, didn't exist. Yes? Okay, how people store food like meat, fish, do you know this? They use the salt, or better if we have spices. Imagine curcuma, curry, I don't know, spices to store with during many months, for example, meat and fish. So the spices were very, ¿cómo decir? Spices se dice, las especias, ¿cómo se dice? Spices, no? Were very important because they allow to store food. So there were expeditions, okay, around the world in this ship to go to specific place and buy all these spices and come back to Europe, okay? But what happened in this journey that maybe lasts 3, 4, 5, 6, 7 months? The diet didn't include anything with vitamin C. Estamos. So what happened after 2, 3, 4, 5, 6, 8 months of navigation? Of course, no vitamin C, no synthesis of collagen, and actually they first start to bleed, but then a lot of sailors died because of scurvy, okay, during this navigation in order to buy the spices that were used to store food. Is a nice story? Yes? Okay. So, in theory, you have to study this in anthropology, but I don't know if you are going to study this. Or otherwise, if you are interested, you can ask the teacher to tell something more regarding this non-genetic disease. Okay, guys, I think that we stop because I'm tired, you are tired, everyone is